Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
When you look at the major diseases we face, few hit as hard or are quite as frustrating to treat as stroke. The impact, socially, emotionally, it's just huge. We're talking permanent disability for many massive long-term costs. And yet, after all these years of research, decades really, we're still working with a tiny toolkit of treatments, and they only work in a very narrow time window.
That really is the tough reality of stroke care today. We've gotten quite good at dealing with the initial blockage, physically pulling out the clot or using drugs to dissolve it. But the real challenge starts after the blood flow returns. That's when this whole cascade of secondary injury kicks off. And frankly, we're still not great at managing that. And it's that secondary damage that often dictates how well a patient recovers in the long run.
Okay, so imagine this. What if a potential answer to tackling that secondary damage wasn't some brand new synthetic drug, but actually a compound found naturally in plants, in some animals, even in tiny amounts in our own bodies? Today we're diving into something pretty astonishing. The idea that dimethyltryptamine, DMT, which most people know as a really potent, fast-acting psychoactive substance, well, it might actually hold a key to protecting the brain right after stroke hits.
It's genuinely remarkable research we're looking at. This deep dive is centered on some groundbreaking work, published relatively recently in Science Advances back in 2025. It came from an international team, researchers from the HN Biological Research Center's Institute of Biophysics and Semmelweis University over in Hungary. And they provided really the first solid evidence showing that DMT can act as a powerful protector for the brain's blood vessels after a stroke.
So, our mission today is to take you right into the thick of what happens during a stroke, that chaotic aftermath, and then show you exactly how this natural compound seems to step in. We really need to explore the specifics, this kind of dual action protective effect DMT seems to have. We'll focus on two critical areas where our current treatments often fall short. First, stabilizing the brain's physical defense system, the blood-brain barrier. And second, calming down that destructive sort of runaway inflammation that explodes after the initial stroke injury. Right, let's start with why stroke is such a difficult beast to tame with drugs designed to protect the brain. We know the immediate trigger is ischemia, lack of oxygen because an artery is blocked. But like you said, the real trouble brews afterwards. Why have so many promising drugs, ones that looked great in the lab, targeting just one specific problem, why have they consistently failed when it comes to actual clinical use?
Yeah, it's a graveyard of failed trials, unfortunately. And the core reason is that stroke damage isn't just about neurons dying from lack of oxygen. The brain has this intricate support system, we call it the neurovascular unit. It's not just the neurons. It includes the tiny blood vessels, the endothelial cells lining them, supportive cells called pericytes, astrocytes, which are like the brain's maintenance crew, and microglia, the immune cells. When ischemia hits, this whole interconnected unit starts to fail almost simultaneously. So trying to fix just one part, like only saving a specific type of neuron, is a bit like trying to patch one tiny hole in a collapsing dam while ignoring the massive pressure building up everywhere else. It just doesn't work.
So in a way, the body's own reaction to the initial injury ends up causing a lot of the long-term damage.
That's exactly it. Once that oxygen supply is cut off, and then often very suddenly restored when we clear the blockage that's called reperfusion, we face two huge interconnected problems almost immediately. The first one is basically vascular failure. The endothelial cells lining the blood vessels get damaged by the lack of oxygen and the subsequent inflammatory signals. And this damage leads directly to a widespread breakdown of that critical physical barrier, the blood-brain barrier.
And when that barrier, that wall, gets breached, what actually happens inside the brain tissue itself?
Well, once it's compromised, you get what's called extravasation.
Yeah.
Things that are normally kept out of the brain, stuff from the blood, like large proteins, albumin is a key one, and other potentially toxic plasma components, they start leaking into the delicate brain environment. This leakage is incredibly disruptive, and it basically signals that the brain's carefully controlled environment is now wide open.
And that breach then triggers the second wave, that destructive inflammatory milieu you mentioned.
Precisely. The brain sees these leak substances as foreign invaders, plus the cells are already stressed. This wakes up the brain's resident immune cells, especially the microglia and astrocytes. They go into high alert, releasing a flood of inflammatory chemicals, things called cytokines and chemokines. This chemical storm causes massive brain-swelling cerebral edema as fluid rushes into the damaged areas. This swelling is dangerous on its own because it increases pressure inside the skull, but it also fuels further damage, expanding the area of permanent injury well beyond the initial spot that lost blood flow. We call this secondary lesion expansion.
Okay, suddenly the limitations of what we can do medically become really stark. Removing the clot quickly is vital, yes, but it doesn't stop this secondary cascade of damage.
Absolutely. Those clot-busting treatments are life-saving, no question, but they have to be given incredibly quickly, usually within just a few hours of the stroke starting. And even when they work perfectly, they don't really address the brain swelling that follows or the ongoing neuron death driven by all that inflammation. We urgently need something that can step in during that chaotic period after the clot is gone, something that can stabilize the brain structure and calm down the immune system at the same time. And that critical need is exactly why this research into DMT is potentially so exciting.
Okay, so the researchers knew they needed a potent agent, something that could hit multiple targets to tackle this really complex, multi-stage disease. Let's unpack how they actually set up the study. Given DMT's nature, you can't just jump into testing it on stroke patients. What kind of models did the Hungarian team use to see if it actually protects the brain?
They took a really smart, two-pronged approach to make sure their findings were solid and relevant. First, and this is crucial, they used an in vivo model testing in a living system. They used rats and induced what's called a transient middle cerebral artery occlusion, or TMCAO. This method is basically the gold standard in labs for mimicking a human ischemic stroke. It involves temporarily blocking the main arteries supplying blood to a large part of the brain, usually for about an hour, to simulate the stroke event itself.
And what about that critical period right after the blockage when the secondary damage really takes hold?
Right, that's key. After the 1-hour blockage, they allowed blood flow to return, that's the reperfusion part, and they monitored the animals for 24 hours. This 24-hour reperfusion window is absolutely critical for testing neuroprotective drugs, because while the initial lack of oxygen causes damage, much of the really destructive stuff, the massive inflammation, the barrier breakdown, the dangerous swelling, actually peaks during these first 24 hours after blood flow is restored. So if DMT could show benefits during this highly damaging phase, it would strongly suggest it has real therapeutic potential.
Okay, that covers the whole animal system. What was the second part of their approach, the more detailed look at the cellular level?
For that, they used a very sophisticated in vitro model. Basically, they built a mini blood-brain barrier in a lab dish. It's a complex setup called a triple co-culture system. They grow 3 essential cell types together: the primary brain endothelial cells that form the vessel lining, the parasites that support them, and glial cells, which include astrocytes and microglia. Then they simulated stroke conditions right in that dish. They used something called oxygen-glucose deprivation, followed by reoxygenation, or OGDR. This mimics the loss of blood supply and then its sudden return, just like in a real stroke and reperfusion scenario. This setup allowed them to really zero in on DMT's direct effects on the barrier cells themselves without the complexity of the whole body systems interfering.
Right, that dual strategy, checking the overall outcome in the animal, then confirming the direct mechanism in the dish, that's really important for showing cause and effect. So the big question, what did they actually find when they gave DMT to the rats experiencing stroke?
The results were really quite striking and statistically very strong. The main headline finding was a huge reduction in brain damage. DMT treatment significantly reduced the size of the cerebral infarct lesion, that's the area of permanently damaged brain tissue. And they didn't just rely on one measurement. They confirmed this using 2 different methods, looking at stained brain slices under a microscope, which is histology, and also using small animal MRI scans. So objective, verifiable proof of less damage.
And did it also tackle that dangerous brain swelling, the edema?
Yes, it did. The DMT treatment also led to a significant reduction in the associated cerebral edema. Given how difficult brain swelling is to treat effectively with drugs after a stroke, this finding on its own is a really big deal.
Now, DMT is known to interact with several systems in the brain, but one receptor often comes up in neurobiology discussions, the sigma-1 receptor. Did they find a connection between DMT's protective effects and this specific receptor?
They definitely investigated that connection very carefully. And yes, they found that the overall protective effect, particularly that reduction in the size of the damaged brain area, was largely dependent on the sigma-1 receptor. How did they show that? They did experiments where they gave the rats DMT together with a compound called BD163, which is known to specifically block the sigma-1 receptor. When they did that, the protective effect of DMT vanished. The blocker basically canceled out DMT's ability to reduce the lesion size. So this strongly pointed to the sigma-1 receptor being a key player, a central mediator for these really crucial neuroprotective benefits of DMT.
Okay, this is where it gets really fascinating, moving from what it does to how it does it. So DMT seems to be acting almost like a biological repair crew, shoring up the brain's physical defenses after the stroke has weakened them. How exactly did the study show DMT stabilizing that blood-brain barrier?
Well, first they looked at the barrier's function in the living animal using a permeability test. They injected a special fluorescent dye attached to a large protein, albumin, it's often abbreviated as FITC albumin, into the rat's bloodstream. Normally, in a healthy brain, that big albumin molecule absolutely should not be able to cross the blood-brain barrier. But after a stroke, the barrier leaks, and the dye gets into the brain tissue. What they found was that DMT administration significantly reduced this leakage. There was much less of the fluorescent dye getting into both the main brain tissue, the parenchyma, and into the cerebrospinal fluid that surrounds the brain. This is really strong evidence that the physical barrier was either better maintained or repaired in the DMT-treated animals.
Okay, so the leaks were reduced. But what's happening down at the molecular level to actually plug those holes? We talk about the barrier like a wall, but it's more like tightly sealed seams between cells, right?
Exactly. The integrity of that barrier relies heavily on specialized proteins that form what are called tight junctions. These proteins literally zip up the space between the endothelial cells lining the blood vessels. Important ones have names like claudins, occludins, and ZO1. A major problem in stroke is that these crucial junction proteins get broken down very quickly. And what DMT seemed to do was protect one of the most critical of these, a protein called claudin 5. They found that DMT prevented the stroke-induced loss of claudin 5, specifically in the tiny brain capillaries. So you could think of DMT as helping to keep the mortar between the cellular bricks intact.
We've heard in other research that it's not just about preserving these proteins, but also stopping the things that actively destroy them. Did DMT affect those destructive elements?
Yes, that's a crucial piece of the puzzle. One of the main culprits that breaks down the barrier structure during stroke is an enzyme called matrix metalloproteinase 9, or MMP9. Think of MMP9 as molecular scissors. When it's released in large amounts after stroke, it specifically snips apart structural proteins like claudin 5, dissolving those tight junctions and causing the barrier to leak. The researchers found DMT did two things here. It helped preserve the claudin 5 protein itself, and it also seemed to block the increase in circulating evidence of this destruction.
What kind of circulating evidence are we talking about? Things they could measure in the blood?
Exactly. They looked in the serum, the liquid part of the blood, and they found that DMT treatment reduced the stroke-induced increase in several key markers. One is the soluble form of claudin 5, basically, the broken-off fragments of the protein floating in the blood. Another was MMP9 itself, those molecular scissors. And they also saw a reduction in another marker called glial fibrillary acidic protein, or GFAP, which often increases when astrocytes, those support cells, are distressed or damaged. The important thing here is that high levels of these three things in the blood, the claudin 5 fragments, MMP9, and GFAP, are known to correlate with worse barrier breakdown and poorer outcomes in human stroke patients. So DMT was effectively reducing these danger signals in the circulation.
And the structural protection, did it also link to how DMT reduced the brain swelling, the edema? You mentioned astrocytes play a role there too.
Yes, it connects back to those astroglial cells. They play a big role in controlling water movement in the brain. A key protein involved in brain swelling, particularly the dangerous type called cytotoxic edema, is a water channel called aquaporin-4, or AQP4. After a stroke, AQP4 expression often goes up, and the protein gets redistributed on the astrocyte surface in a way that lets too much water flood into the brain tissue. What DMT did was inhibit this stroke-induced increase and abnormal redistribution of AQP4, especially around the capillaries. So by helping to stabilize how AQP4 functions, DMT helps manage the water balance, which is critical for reducing that dangerous swelling and pressure buildup.
And let's not forget the results from the lab dish model, the in vitro part. Did that confirm these direct barrier effects?
It absolutely did. In that triple co-culture model where they simulated stroke conditions using oxygen and glucose deprivation, DMT again showed its protective effects. It significantly blocked the increase in permeability across that artificial barrier, meaning less simulated leakage. This provides really strong support for the idea that DMT has a direct local effect on the cells of the barrier itself, helping them resist the stroke-induced damage.
Okay, so DMT is acting like a structural engineer, reinforcing the castle walls of the blood-brain barrier. Now let's look at its other major role, acting like a firefighter, calming down the immune response that goes haywire after a stroke. How does DMT manage to quell this inflammation, both within the brain and systemically?
Let's start inside the brain with the resident immune cells, the microglia. Normally, microglia are like sentinels surveying their environment, but after a stroke, they get activated and often shift into a pro-inflammatory, potentially damaging state. The study found that DMT significantly suppressed this harmful activation of microglia.
How could they actually measure that? How do you quantify whether immune cells are activated in a damaging way?
They used a really neat technique, looking closely at the cell's shape, their morphology. Microglia physically change shape depending on what they're doing. When they're in a healthy, resting surveillance state, they tend to have lots of fine branches.
Yeah.
They call this a highly ramified shape, which they classified as cluster 0. But when they get activated after injury, they often pull in these branches, becoming less ramified, sometimes almost amoeba-like. This less branched shape, like the one they call cluster 2, is often associated with a more destructive, pro-inflammatory function. What DMT treatment did was cause a clear shift away from that reactive, less ramified cluster 2 shape, and it helped preserve the healthier, highly branched cluster 0 morphology. Essentially, DMT seems to encourage the microglia to stay in, or return to, a state that's more focused on cleaning up damage and promoting repair, rather than just fueling inflammation.
Wow, that's quite profound. It's not just turning down the volume on inflammation, it's actually influencing the type of response the brain's own immune cells are mounting, steering them towards healing. What about the chemical signals, that flood of inflammatory molecules we talked about?
DMT tackled that too, both in the animal models and in the cell culture experiments. It acted like an effective dimmer switch. DMT treatment significantly reduced the release of several key pro-inflammatory cytokines. These are molecules like tumor necrosis factor alpha, TNF-alpha, interleukin-1 beta, IL-1 beta, and interleukin-6, IL-6. These cytokines are like the alarm bells that recruit more immune cells and generally ramp up the inflammatory cascade. DMT turned down those alarms, both in the brain endothelial cells and in immune cells circulating in the blood.
And this calming effect wasn't just contained within the brain, right? You mentioned changes in the blood too, suggesting a system-wide effect.
Absolutely. When they looked at the serum, the liquid part of the blood flowing through the whole body, they saw a really significant shift in the balance of these signaling molecules. DMT significantly lowered the levels of those damaging pro-inflammatory cytokines we just mentioned. It also reduced key chemokines like CXCL1 and CXCL10. Chemokines are the signals that act like come here calls, recruiting more inflammatory cells like neutrophils from the general circulation into the injured brain, which just adds fuel to the fire. DMT reduced those recruitment signals.
But perhaps even more exciting is the other side of the coin, not just reducing the bad stuff, but actually increasing the good stuff, the molecules associated with healing and recovery.
Yes, this is where it gets really interesting for potential long-term benefits. DMT didn't just suppress inflammation. It actively promoted an anti-inflammatory and pro-recovery environment. Specifically in the serum, it significantly increased levels of important anti-inflammatory cytokine called interleukin-10, IL-10. And perhaps most critically, it dramatically boosted levels of a protein called brain-derived neurotrophic factor, or BDNF. BDNF is incredibly important. High levels aren't just linked to better neuron survival. In human stroke patients, higher BDNF levels are consistently associated with better overall recovery, improved neuroplasticity, the brain's ability to rewire itself, and a lower risk of developing cognitive problems long term. So DMT appears to be actively flipping the switch from a destructive inflammatory state towards one that supports repair and regeneration.
Okay, so we've built this picture of DMT as this incredibly versatile agent, a pleiotropic molecule, hitting multiple targets at once. It's fixing the barrier, calming the immune system, like a biological Swiss Army knife. And we know from the earlier results that the sigma-1 receptor seems crucial for the main protective effect, reducing the overall damage. But here's a question. Researchers have tried targeting the sigma-1 receptor before with other drugs, often specifically designed agonists, and those haven't always translated well into successful stroke therapies in humans. Why might DMT, acting through sigma-1 but also doing other things, be different? Why does its multi-target nature maybe give it an edge?
That's a really important point and gets to the heart of why this study is so compelling. The fact that some previous single-target sigma-1 drugs didn't pan out clinically suggests that just hitting that one receptor might not be enough to overcome the sheer complexity and chaos of what's happening in the brain after a stroke. DMT seems to succeed, potentially, because while it does strongly engage the sigma-1 receptor, it's simultaneously doing other beneficial things, particularly stabilizing that barrier very quickly. We can see this multifaceted action really clearly when we look again at those experiments where they use the sigma-1 blocker, BD1063. That helps tease apart which effects absolutely require sigma-1 and which might involve other pathways.
Right, so let's break that down. Which specific benefits of DMT were clearly shown to depend on the sigma-1 receptor pathway?
The majority of the core therapeutic effects seem tightly linked to sigma-1. So that big one reducing the overall size of the stroke lesion, the infarct volume that was blocked by BD1063. Also, the beneficial effect on astrocytes, specifically mitigating that dysfunction of the aquaporin 4 water channel involved in swelling, that also seemed sigma-1 dependent. And the powerful anti-inflammatory actions reducing those key pro-inflammatory cytokines like TNF-alpha and IL-1 beta, and also preserving that healthy branch shape of the microglia, all of those effects were significantly reduced or completely blocked when the sigma-1 receptor was inhibited. So it's clear that the sigma-1 pathway is the main engine driving this complex neuroprotection and the shift away from inflammation.
But, and this is the really interesting nuance, the researchers found that some protective effects still happened even when that sigma-1 engine was switched off by the blocker, which effects seem to be at least partly independent of the sigma-1 receptor.
This was a fascinating finding. The most significant one was related to the blood-brain barrier itself. Remember how DMT reduced the leakage of that fluorescent dye into the brain in the animal model?
Right.
Where that specific effect, the reduction of barrier permeability in vivo, was not fully reversed when they blocked the sigma-1 receptor. DMT still provided some barrier protection even without sigma-1 being fully active. Furthermore, the reduction they saw in those circulating chemokines in the blood, CXCL1 and CXCL10, the ones that recruit more immune cells, that reduction also seemed to persist even when BD1063 was present.
Okay, that is a critical piece of information. It suggests DMT has this sort of built-in secondary mechanism for stabilizing the blood vessels that runs alongside or maybe complements the main sigma-1 pathway. Do we have any idea what these other mechanisms might be? What else could DMT be doing to protect the barrier directly?
The paper doesn't definitively pinpoint these secondary mechanisms, but based on what we know about DMT's broader pharmacology, we can certainly speculate. DMT is perhaps most famous for its strong interaction with serotonin receptors, especially the 5-HT2A receptor, which is linked to its psychedelic effects. But it's known to interact, albeit sometimes more weakly or transiently, with a whole range of other targets to potentially other serotonin receptor subtypes, maybe certain ion channels, or even interacting directly with the cell membranes themselves, influencing their fluidity or organization. It's plausible that these alternative interactions, perhaps happening very rapidly, could be responsible for that immediate, robust stabilization of the endothelial cells and the barrier integrity that persists even when sigma-1 is blocked.
So the picture that emerges is sigma-1 is crucial for the longer-term programming, the anti-inflammatory shift, the neuron survival signals, but there might be this separate, perhaps faster-acting pathway that DMT uses for immediate damage control, just physically holding the barrier together.
Exactly. And in the context of acute stroke, where every minute counts, having that kind of multi-pronged attack could be a massive advantage. It means the drug isn't relying on just one mechanism. It's hitting the problem from multiple angles simultaneously, offering a level of robustness and maybe redundancy that a drug targeting only a single pathway might struggle to achieve in such a complex and rapidly changing disease state. This pleiotropy, this ability to do several beneficial things at once, is really what makes DMT stand out as a potential therapeutic in this context.
Right. We've established this compelling picture. DMT with its dual action, protecting the vessels, calming the inflammation. But we do have to tackle the elephant in the room, so to speak. DMT is, in many parts of the world, a strictly controlled substance, known primarily for its intense psychedelic effects. How do we even begin to think about using a compound like this in an emergency clinical setting, like right after someone has a stroke? What are the practical hurdles?
That is absolutely the critical question for translating these fascinating lab findings into something that could actually help patients. There are really two main lines of thought or approaches here. First, and perhaps most likely for widespread clinical use, is the development of non-hallucinogenic analogs or derivatives of DMT. It's chemically possible to modify the DMT molecule slightly. The goal would be to keep its strong binding affinity for the sigma-1 receptor, which seems to drive the crucial neuroprotective and anti-inflammatory effects, while significantly reducing its ability to activate the serotonin 5-HT2A receptor, which is primarily responsible for the psychedelic experience. There's already active research happening in pharmacology aimed at creating precisely these kinds of safer, non-psychedelic compounds that still target sigma-1 effectively.
Okay, modifying the molecule is one route. What's the second approach? Does it involve using DMT itself, but maybe differently?
The second approach considers the context of use. Even if the original DMT molecule were used, we have to remember the situation. We're talking about acute stroke intervention. The therapeutic window here isn't about recreational use. It's likely a very short period, maybe hours or just a couple of days right after the stroke occurs and blood flow is restored. The primary goal is maximizing brain cell survival during that critical initial recovery phase. It's also possible that the doses needed to achieve a rapid neuroprotective effect on the blood vessels and inflammation might be different, perhaps lower, than the doses required to induce a full-blown psychedelic experience. The idea would be to use it as a very specific, time-limited pharmacological tool to help stabilize the patient and protect the brain right when it's most vulnerable.
The researchers themselves seem very focused on this dual action as the key benefit. One of the co-first authors, Judith Vai, was quoted emphasizing this. She said essentially that current stroke treatments are very limited, and that DMT's ability to both protect the blood-brain barrier and reduce brain inflammation offers a new, complex strategy that could work alongside or complement the treatments we already have. It's not necessarily about replacing current methods, but enhancing them.
That's absolutely the perspective. DMT, or a derivative, isn't seen as a magic bullet replacement for, say, clot removal. It's viewed as a potentially crucial addition to the treatment pathway. You can imagine a scenario. A patient comes in, they successfully undergo thrombectomy, the clot is cleared. But we know that reperfusion injury is coming. That's when you might administer DMT or a related compound to shore up the blood vessels as blood flow returns, to dampen that inflammatory surge, and importantly, to boost those factors like BDNF that set the stage for better long-term healing and recovery.
So it bridges that gap between the immediate clot removal and the longer-term rehabilitation phase, potentially making that rehab more effective. What are the actual next steps then in terms of clinical trials? Is this moving towards human testing?
Yes, these strong preclinical results have definitely spurred clinical development. There are already phase 1 trials underway. These initial trials are mainly focused on safety, figuring out the right doses, and how the compound behaves when given intravenously to healthy volunteers, standard procedure for any new drug candidate. But crucially, the research team behind this work mentioned they are actively designing a phase 2 trial. The specific focus for that phase 2 study will be to look at DMT's potential as an enhancer of neuroplasticity, the brain's ability to adapt and rewire. They want to investigate directly whether giving it to patients shortly after an acute stroke actually improves their long-term recovery outcomes.
And that potential to boost neuroplasticity really feels like the ultimate prize here. It's not just about keeping brain cells alive immediately after the stroke, but about creating an environment where the brain can actually heal and regain function more effectively down the line. By increasing that brain-derived neurotrophic factor, BDNF, and creating a less inflammatory, less hostile environment, DMT seems like it could be preparing the brain for the hard work of rehabilitation and rewiring that needs to happen.
It really connects the dots, doesn't it? It links what we know about this molecule's effects on brain function and maybe even consciousness with this newly discovered fundamental ability to protect the brain's physical structure and modulate its immune response at a critical time. It paints a picture of DMT as potentially a very versatile tool useful for acute protection right after the injury, and maybe also for promoting better functional recovery in the weeks and months that follow.
So, wrapping up our deep dive today, there are really 2 key takeaways for you, the listener, from this fascinating research. First, the core finding. DMT significantly reduces the amount of brain damage and dangerous swelling in experimental stroke models. That's the headline result. And second, the how. It seems to achieve this through a powerful 2-pronged approach. It reinforces the physical blood-brain barrier, protecting proteins like claudin-5 to keep it sealed, and simultaneously it shifts the body's inflammatory response away from damage and towards protection, importantly boosting levels of brain-derived neurotrophic factor, which is vital for long-term brain repair and plasticity.
And thinking about this really leaves us with a profound final thought, doesn't it? Dimethyltryptamine is, after all, a molecule that occurs naturally, albeit in small amounts, within the human brain and body. If our own biology contains compounds with such potent multi-target protective abilities against one of our most devastating and complex diseases, a disease like stroke, which we still struggle so much to treat effectively, it really makes you wonder. What other endogenous molecules, what other natural compounds produced within us are just waiting to be properly understood, waiting to be potentially leveraged for therapeutic benefit against stroke or maybe other challenging conditions? It's a reminder, perhaps, to keep questioning, keep exploring the incredible, largely untapped potential hidden within our own neurobiology and the natural pharmacy inside us all.
Thanks for tuning in to the Health Headliner podcast by Two Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.